Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, we are already in a continuation of the chapter that is photosynthesis. In previous class, we have already talked about the different experiments which was done by different scientists to prove the essentiality of different factors in the photosynthesis. Now we are going to discuss from here that how the photosynthesis take place actually in plant. But before that, we have to discuss where the photosynthesis takes place, that is the site of photosynthesis, and what are the machinery which is required for photosynthesis, that is the special pigments. The pigments are required here. So where these pigments are located and which pigments are involved, that all thing we have to discuss here. Okay. So if we talk about the first reactions of the uh, photosynthesis, then we know that there is a 6 CO2 when react with 12 water molecules in the presence of sunlight and pigments is going to give rise to C6H12O6 plus 6O2 plus 6H2O. Here CO2 is going to consume and O2 is going to be evolved. So if I link this reaction, dear student, suppose in the first phase the water is getting used and this oxygen and okay, oxygen and OH2 are produced from this oil. This is the first phase, and this first phase is known as light reaction. So whole photosynthesis is divided into two phases. This is the first phase, which is known as light reaction or Hill reaction. And in this light reaction or Hill reaction, this is the first phase. And the second phase where the CO2 are used and glucose is produced. There is the second phase, and the second phase is known as a dark reaction or Blackman reaction. Now, dear student, in light reaction or Hill reaction, where the H2 is used and oxygen and water are produced, in that light reaction, we will study non-cyclic and cyclic photophosphorylation. These two processes we are going to study. And in this non-cyclic and cyclic photophosphorylation, you will see that and we will study that the synthesis of ATP and NADPH2 occurs. So ATP and NADPH2 are synthesized. So these are going to synthesize in a light reaction. Now this particular products of the light reaction, that is ATP and NADPH2, are used in dark reaction where CO2 fixation occur and glucose formation will take place. Now in dark reaction, we will study C3 cycle, C4 cycle, and Chem cycle or pathway. These are ultimately involved in glucose synthesis by CO2 fixation. So these things actually we are going to study in the process. So light reaction, dark reaction, two phases are there in whole photosynthesis. In light reaction, ATP and NADPH2 and are synthesized, and this light reaction is directly dependent on the sunlight. Okay, but the products of this light reaction are ATP and NADPH2, which are used in dark reaction. And in dark reaction, it's Blackman reaction. The C3, C4, and Chem cycles will going to occur, and that will carry out a glucose synthesis. So this is the theme of the chapter. What we are going to study in the process. Now, where this light reaction and dark reaction takes place, what is the site of photosynthesis that we have to discuss. So generally we say that green leaves or green parts of the plant are the site of photosynthesis. Obviously green areas or green parts are the particularly involved in photosynthesis, but it also occur in other green parts of the plant. Apart from green leaves, also occur in green parts of the plant. And in plant cell, if we talk, there is a mesophyll cell. They have a chloroplast. Okay, and this cell containing chloroplast are involved in particularly photosynthesis. So these are the chloroplast, okay, and they particularly perform photosynthesis. So these are the mesophyll cell, and mesophyll cell have this chloroplast. These are round box showing the chloroplast. That chloroplast carry out this particular. So there is a clear-cut division of labor within the chloroplast. And in chloroplast, this photosynthesis occurs in the green parts. Then what is this photosynthetic parts? So photosynthetic parts or a green parts, other green parts of the plant, green leaves and other green parts of the plant where the photosynthesis takes place. First is the leaves. Apart from leaf, you will see that in young stem also photosynthesis can occur. Apart from this, calyx is there. Calyx is the group of sepal in the flower that also particularly carry out photosynthesis. This one you can see. Okay. Now, apart from this, the skin of raw fruit, green raw fruit, where also photosynthesis can occur. Okay, so these are the site of photosynthesis where green parts of the plant where photosynthesis occur. And in green parts, there is a chloroplast where the photosynthesis takes place. So if we talk about the structure of chloroplast first, inside the chloroplast there is a membrane system consisting of a grana and stroma lamella and the fluid stroma. And you will see that there is a clear cut division of labor within the chloroplast. We will understand by studying this structure. Okay. The membrane system is there. Chloroplast is membranous organelle, okay, within the plant cell. So it is a membrane bound and membrane system is there that is responsible for trapping the light energy and also for synthesizing ATP and NADPH2. Means for the light reaction, the membranes are important in this one. So this is the structure of chloroplast. Okay. Dear student, if we talk about the structure of chloroplast, if we talk, it is a double membrane bound. It is a double membrane bound organelle. So this is the outer membrane, this is the inner membrane, these two membrane are covering the chloroplast. Within the chloroplast, you will see these structures which are known as thylakoids. Okay, you can see this is the single one. This one is the thylakoid. Okay, these all are the thylakoid. But if we talk this thylakoids, then this thylakoid are piling up and they are after piling they are forming a granum. So this thylakoid are known as grana thylakoid. They are known as grana thylakoid. Or we can say grana lamella. Thylakoid are also named as lamella. So we can call this grana lamella. Whereas this is the connecting two granum. This is the one granum, this is another granum. These two granum are connected by this thylakoid, and then this thylakoid is known as stroma thylakoid or stroma lamella. And this is the stroma. Now in stroma, dark reaction occur and on membranous system, light reaction will take place. So light reaction occur in the particularly this membranous system, and in the thylakoid membrane you will see the pigment. That pigment are involved in absorption of light. Okay, and they after absorbing light they synthesize a ATP and NADPH2 in the light reaction. After this ATP and NADPH2 are used in this stroma and dark reaction will take place here. Okay, so this way this process is going to occur. So now this is the what a structure of chloroplast. So they have membranous system and they have a stroma system. So in stroma, there are enzymatic reactions which incorporate the CO2, means fix the CO2 into the plant and lead to the synthesis of sugar, which in turn form a starch. Okay, so that is a dark reaction. So what we have studied, light reaction takes place on the membranous system. Whatever the pigment involved in membranous that are involved in a light reaction. So membranous system is responsible for carrying out a light reaction and synthesis of ATP and NADPH2. But dark reaction is carried out in a stroma and it fixes the CO2 and synthesizes sugar. So the former set of reactions, since they are directly light driven, means light reaction is directly light driven, they are called a light reaction. Whereas the latter are not directly dependent on the light reaction, but they are dependent on the products of light reaction, that is ATP and NADPH2. So they are called by convention dark reaction. Actually, dark reaction here does not mean it occur in dark, but it is dependent on the products of light reaction. Because of this, we are calling it as a dark reaction. So, however, this should not be taken to mean that they occur in darkness or they are not light dependent. Okay, That is not meant by the dark reaction. Now, indirect reaction or light reaction, where they take place, that we have discussed already about the structure of chloroplast. Now, when we talk about the structure of chloroplast and when we talk about the position of the light reaction and dark reaction, now the important thing which is involved in absorption of light to carry out a light reaction, that is the pigments. And these pigments are found in, particularly we can say, the chloroplast thylakoid membrane in group, and this group is known as photosystem. Okay, this group is known as photosystem. So first we have to talk about what is photosystem or pigment system. Now photosystem is nothing but a group of pigment, because in thylakoid membrane or in chloroplast, the pigments are not going to found in isolated or singly, they will not occur, they will occur in group. So photosystem or pigment system, if we talk, it is a group of pigments. It is a group of pigments. Okay. Now this photosystem or pigment system, if we talk, it is a group of pigments, and this group of pigments, if we talk, they are going to occur here. Now this group of pigment or photosystem or pigment system is there. Each photosystem has the same structure. So if we talk about a structure, this is the structure of photosystem. And in each photosystem, you will see that there is a one reaction center. This is the reaction center, which is always chlorophyll A. Okay. And this is the primary electron acceptor of this reaction center or photosystem. Okay. And these are forming a reaction center. Now this all somewhat light greenish are in color, they all are the accessory pigment or antenna pigment molecule. So antenna pigment molecule are nothing but accessory pigment. They are accessory pigments. And accessory pigments are the antenna pigment molecules. Okay. Now this accessory pigment, if we talk, they are the group of pigments, and this is the particularly we can say the reaction center. Now what this accessory pigment do? They accept the light energy, then transfer this light energy ultimately when pigment by passing to the another pigment ultimately to the reaction center. Now in reaction center you will see the chlorophyll, and chlorophyll have a magnesium. From the magnesium of this chlorophyll, the ionization will take place and electron will get transferred to the what or primary electron acceptor. And from that they will be transferred again to the further electron carriers. So means this group of pigments or photosystem has two components. One is reaction center, that is chlorophyll A, and another one is the, we can say, antenna molecule. And this antenna is also known as accessory pigments. Antenna has also accessory pigment or light harvesting complex. Light harvesting complex. Okay, so these all are there in a particular photosystem, but photosystem has reaction center and accessory pigment. Accessory pigment has a function or to pass on this particular energy of light to the ultimately reaction center, and in reaction center electron excitation will take place for the magnesium of the chlorophyll A, and electron will pass to the this primary electron acceptor. So this reaction center and accessory pigments are combinedly form one photosystem. Okay, and there are several photosystem are there in the particularly membrane, but this photosystem are of two type. This photosystem are of two type. based on discovery. Based on discovery. One is PS1, that is photosystem 1, and another one is the PS2. Now, when we talk about PS1 and PS2, PS1 is a photosystem 1, and this PS2 is photosystem 2. PS1 or photosystem 1, if we talk, PS2 or photosystem 2, the PS1 its absorption maxima. It is also a group of pigment. It is what discovered first, so it is named as PS1, and it is identified that its absorption maxima is 700 nanometer. Means the maximum wavelength which they can absorb, this photosystem 1 that can absorb, it is 700 nanometer. And PS2, its absorption maxima is 680 nanometer. Its absorption maxima is 680 nanometer. Now, means it can absorb wavelength between, or we can say it can absorb a wavelength from greater than 680 to 700 nanometer. And this PS2 can absorb wavelength from 400 to 680 nanometer. Okay, so this is the absorption. There is a difference in this particular absorption segment. Now its absorption eczema is 700 nanometer, so means its particular reaction center has a 700 nanometer at which it get excited, whereas in PS2 the absorption maxima is 680, means 680 nanometer chlorophyll will be at the reaction center. Okay, now this is the what PS1 and PS2 do. Now where they are located, that we have to discuss. So next we are going to talk about the location of PS1 and PS2. When we talk about the location of PS1 and PS2, suppose this is the granathylakoid. Okay, and this is the stromathylakoid. So this is the granathylakoid and this is the stromathylakoid and this is their membranes. And this is granathylakoid. Now, dear student, where this photosystem 1 and photosystem 2 are located, that we have to discuss. So be remember, here you will see the presence of PS1. Here also you can see the presence of PS1. Here also you can see the presence of PS1. Here, all this point you will see the presence of PS1. Here also, these all are PS1. Means PS1 is located on where? Free surface. On the free surface, see, this is the common surface. That common surface you will not see the presence of PS1, but on the free surface you will see the presence of PS1. Means PS1 is located on the non-appraised or free surface of the grana and stroma thylakoid. Whereas the common surface, wherever you will see the common surface, at this common surface you will see the presence of PS2. Means the surface which is placed between two thylakoids, there you will see the presence of PS2. So these are the PS2. Okay, these are the PS2. These are the PS2. Okay, so here now it is clear that PS1, PS1 is located on non-oppressed non-oppressed or free surface or parts of grana and stroma thylakoid, whereas PS2 is located at oppressed or common surface of grana thylakoid. Means PS1 is the only photosystem which can be present on the membrane of both grana and stroma thylakoid. But PS2 will be located on the only grana thylakoid because the common surfaces will be seen on the granum only or on the grana thylakoid. And that is the thing where you will see this particular location of PS1 and PS2. Now, these two photosystem operate simultaneously and their operation activation requires 700 nanometer and 1600 nanometer radiation as we have discussed. But this all photosystem ultimately have what? Pigments. They ultimately have what? Pigments. So we have to understand about the pigments first. So how many pigments are involved in photosynthesis? That is our next question. And which pigments are involved? That is our next question. So chlorophyll A, chlorophyll B, xanthophyll, and carotenoids are the different pigments which are involved here. Chlorophyll A, chlorophyll B, xanthophyll, carotenoids. These all are particularly involved here in the photosystem. So how these pigments are first isolated, that is we have to understand. So for that, what we take a leaf tissues in a one by and we crush it by a mortar and pestle. Then we isolate this particular. We take this leaf tissue and we isolate, extract it, and we carry out a paper chromatography, and we obtain a picture that is known as chromatograph. So after paper chromatography, we will understand on a chromatograph that there is a different pigments are found here. So now the question is, have you ever wondered why and how there are so many shades of green in leaves even in the same plant? So the question is because there are so many pigments in the leaves and the different type of pigments are there. So these pigments are separated by paper chromatography as we have discussed, and the chromatographic separation of the leafy pigment shows that the color that we see in the leaves is due to the is not due to a single pigment but is due to the four different pigments. And the four pigments which are found in the leaf, and these four leaf pigments are chlorophyll A, which is bright green or blue green in color. Another one is the carotenoid, which is, we can say, yellow orange, xanthophylls, which are yellow, and chlorophyll B, which is yellow green. Okay, so these four pigments are mainly occur in a leaf, and they are the leaf pigments. So this is the, you can see the chlorophyll A, which is bright or blue green color, that gives this particular coloration. Then carotenoids we have discussed, then xanthophyll we have discussed, and this chlorophyll B we have discussed, that yellow. Because of this particular beautiful picture which arises in the leaf, this is due to these different pigments. So now these pigments have been separated. This is a chromatograph on which you will see the position of different pigments, where keratin are at the upper post, and to the lower xanthophyll will be seen, then chlorophyll A and chlorophyll B. Okay. Now we have to discuss all this pigment in detail, that we will discuss here. So now let's discuss about the different pigments in detail. Okay. So if we classify the pigments, and if we discuss about the type of pigments, type of pigments, there are specifically three types of pigment. One is chlorophylls, another one is the carotenoids, and another one is the phycobilins. These three types of pigments are there. So pigments are classified into three groups. We are going to discuss each group one by one. So first we are going to discuss chlorophylls. Okay. Now when we discuss about the chlorophylls, in chlorophylls, dear student, they are a green color pigment and they occur inside chloroplast. Obviously, when we talk about the chlorophylls, they are green colored pigments, and these chlorophylls occur inside chloroplast. Now, these pigments are lipid in nature. These pigments are lipid in nature. Apart from this, they are insoluble in water and soluble in organic solvent. They are soluble in organic solvents. So these are the characteristic feature of these different chlorophylls. They are lipid in nature. They are lipid molecules. So because of this, they are insoluble in water, but they are soluble in organic solvents. Now these chlorophylls are of different type, and if we talk about the types of chlorophylls, these chlorophylls are of different type, like chlorophyll A, B, C, D, E. Apart from this, you will see the some bacteriochlorophyll. This bacteriochlorophyll is also known as bacteriopurpurin. We will discuss about the chlorophyll A, B, C detail. D and E are not of important, but we remember this bacteriochlorophyll or bacteriopurpurin, which we call, that is a purple color pigment. This is purple color pigment, and the empirical formula or molecular formula of this is C55S74 O6N4Mg. Means they have a carbon, hydrogen, oxygen, nitrogen, and magnesium. And This particular chlorophyll or purpurine, as we have discussed in the biological classification, they occur inside purple bacteria. That can be purple sulfur bacteria or purple non-sulfur bacteria. Apart from this, another which is found in bacteria, that is the bacterioviridin. This bacterioviridin is also known as chlorobium chlorophyll. And this chlorobium chlorophyll, if we talk, that is a green color pigment, and that occur inside green bacteria. Okay, so that is the what a particularly different types of chlorophyll. Now in this, the chlorophyll A is too much important for us. We will understand how chlorophyll A differ from B, and one important point with respect to chlorophyll C. D and E are of not much importance, so we will not discuss that. So let's discuss chlorophyll A first. If we talk about chlorophyll A, it is a bluish green or bright green pigment, and this chlorophyll E with the molecular formula, that is C55 H72 O5 N4 Mg, means they have 55 carbon, 72 hydrogen, 5 oxygen, 4 nitrogen, and 1 magnesium molecule. Now this chlorophyll A is considered as universal photosynthetic pigment. It is a universal photosynthetic pigment. Now the question is, why it is considered as universal photosynthetic pigment? Because it is present in every photosynthetic organism except eubacteria. It is known as universal photosynthetic pigment because present in it is present in every group of photosynthetic organism or every photosynthetic organism except eubacteria. Apart from this, this chlorophyll A is also considered as primary photosynthetic pigment. Now here also question is why it is known as primary photosynthetic pigment, because the primary reaction which involve the conversion of light energy into the chemical energy, that is light reaction, is mediated by chlorophyll A molecule because it acts as a reaction center. So in reaction center, when chlorophyll A is present, it converts light energy into chemical energy. Now apart from this, chlorophyll A is also considered as most abundant photosynthetic pigment. Most abundant photosynthetic pigment. Okay, so this is the what is a chlorophyll A. It is the most important pigment, it is universal pigment, it is a primary pigment, most abundantly found. Now what is the structure and how it is synthesized, that we have to discuss. So first we talk about the synthesis. Synthesis of chlorophyll A. Synthesis of chlorophyll A, if we talk, there is glycine plus succinyl coenzyme A. Now, when glycine and succinyl coenzyme A acting as a precursor, they react, and by reaction of these two, they are going to form a one substance which is known as protochlorophyll, or we can say protochlorophyllidase. Protochlorophyllidase. So glycine react with the succinyl coenzyme A and form protochlorophyllidase. Now, in the presence of light, it is going to form a chlorophyll A. So chlorophyll A is synthesized in the presence of light and from the glycine and succinyl coenzyme A as a precursor. So this is the most important question that you will get repeatedly. That is, what are the precursor of this chlorophyll A? So these are the glycine and succinyl coenzyme A. Now here, three minerals are also required during this. Three minerals, that is magnesium, iron and nitrogen, are essential for chlorophyll synthesis. Are essential for chlorophyll synthesis. And here, for this synthesis, light is compulsory. Light is compulsory for chlorophyll synthesis. For chlorophyll synthesis in angiosperm. Okay, so this is the synthesis. Now we are going to talk about the structure. When we talk about the structure of chlorophyll A, it has a tadpole look like structure. It has a tadpole like structure. And in tadpole like structure, it has two component. It has a two component. One is porphyrin head and another one is the phytol tail. Now this porphyrin head, if we talk, and this is the phytol tail, if we talk, this phytol tail has a C20S39OH. So if we remove this 20 carbon and 39 nitrogen and oxygen, the porphyrin head will remain in the structure. So now this porphyrin head, if we talk, that is hydrophilic, and acidic in nature. That is acidic in nature. And if we talk about a phytol tail, it is hydrophobic. It is hydrophobic and alcoholic in nature. It is alcoholic in nature. And this tail remain embedded in lipid bilayer of thylakoid membrane. So it is known that through tail, chlorophyll A attach to thylakoid membrane. Now we have to understand that how this chlorophyll actually look, what is porphyrinate, what is phytol tail, how this structure look, that we have to understand very carefully. Okay, so if we discuss the structure of chlorophyll A, the structure is somewhat like this. Let's see the structure. So in such structure, for framing a structure, first we have to understand that we remember the magnesium is in center and it linked with four nitrogen through two coordinate bond and two covalent bond. Okay. Now apart from this nitrogen are attached to two carbon each. They are attached to two carbon each, and these are also further attached to the two carbon, and these two carbon are also linked to each other. Okay. Now they have first pyrrole ring. This is second pyrrole ring. This is third pyrrole ring, and this is fourth pyrrole ring. These are known as pyrrole ring, which are consist of one nitrogen and four carbon. Now these four pyrrole ring are attached to each other by the methane bridges. So here you can see this is attached with the carbon and this carbon is attached with the hydrogen. So this valency of four carbon is satisfied for this carbon as well as this carbon. Now this carbon has a particularly we can say the three valency. So to satisfy that, if we make a bond here, that going to satisfy the valency of this carbon. Now here this carbon has a particular valency. Now we remember here this is a methane bridge. Now here also there is a methane bridge. Okay. Now here also this is a methane bridge, and we are putting the double bond on the lower side. Okay. Here also this is the methane bridge. Okay. Now this carbon it attached to the through C double bond O. And this is the fifth pyrrole ring, and this CH is associated with the C double bond O and OCH3. That is OCH3. Now here, dear student, this is the first pyrrole ring, second pyrrole ring, third pyrrole ring, and fourth pyrrole ring, and this is the fifth pentacyclic ring is there. Now first we have to make a valency clear here. Okay, and to particularly accomplish with the valency, we remember that in this is the whole pyrrole head. Now this carbon of each pyrrole ring which are peripheral, they are named like this. This is the first carbon, this is second carbon, this is third carbon, this is fourth carbon. Okay, they are labeled like this. Let's make mark with the other color. This is the first carbon, peripheral carbon we are going to mark. Third, fourth, fifth, sixth, seventh, and eighth. Now, eighth carbon, you will see the attachment of the phytol tail. Okay, phytol tail is attached here at the seventh carbon. Now, apart from this, this is the whole is the porphyrin head. This is the porphyrin head. Now, if you want to particularly add the groups, different group, it is known that there is a methyl group attached at the 1, 3, 5, 8 carbon at first, third, fifth, and 8 carbon. Methyl group is attached. So if attach the methyl group here, at this part, at this part, and at this part. Apart from this, the vinyl group is at the second carbon. At second carbon, vinyl group, that is CH bond, double bond CH2 is attached, while at fourth carbon, ethyl group, that is CH2 CH3 is attached. So 1358 we have added already methyl group. Now at the second carbon we have to add the ethyl group, that is a CH double bond CH2. Okay. And at the fifth carbon, at the fourth carbon, we have to add a ethyl group, that is CH2 CH3. Okay. Now we have to satisfy the valency. So here this carbon has a 4 valency, here 1, okay, here 2. So this nitrogen has 3 valency, so this will be here, so it will be 3 valency will be completed. Here carbon has 4 valency, it cannot bond for, because this carbon's valency are completed, so here the double bond will be here. The valency of this carbon satisfied, valency of this carbon is also satisfied. Okay. Now this carbon CH and CH2, that is also satisfied. Now here the double bond getting satisfied. Here double bond is there, that is satisfying the valency of this carbon. Nitrogen has 3 valency, that is satisfied. This carbon has a 4 valency and this carbon has a 4 valency. So here if we put a double bond here, it is satisfying here double valency. Then if we put a here double bond, then it will be getting the satisfied this valency of these two carbons. Okay. And this way the valency getting satisfied. Now this fifth ring, that fifth ring is known as cyclopentanone ring. Okay, this way that this is the whole chlorophyll A structure. Now at second carbon, we remember this is the vinyl group, and at the third carbon you can see this is having a particularly ethyl group. Okay, now this is the chlorophyll A structure and how chlorophyll A look, that all thing we have discussed. Now the empirical formula is also we have discussed, that is a C55 S72O5 and 4 Mg. Now this is the structure of chlorophyll A. Now chlorophyll A get modified and we can form a chlorophyll B. So chlorophyll B is just somewhat modification of chlorophyll. It is chlorophyll A like. So if we talk about the chlorophyll B, chlorophyll B, chlorophyll B is a yellow green pigment. Chlorophyll B is a yellow green pigment and it has an empirical formula, that is C55 S70O6 N4 Mg. Okay, and chlorophyll B is considered as it is structurally similar to chlorophyll A. Structurally similar to chlorophyll A, except it has CHO group in place of CH3 at third carbon or position of second pyrrole ring. Okay, if we replace this CH3 at third carbon of second pyrrole ring with the CHO, then it get modified into the chlorophyll B. So if you want to understand that, you can see this is the structure we have made. This is the second pyrrole ring in this one. Third carbon is this. So this we are talking about this carbon, this CH3. If this CH3 we replaces with the CHO, it is going to become a chlorophyll B. Apart from this, there is another chlorophyll which is we call chlorophyll C. Now, one peculiarity of chlorophyll C need to be remembered, and that is it lack phytol tail. means phytol tail is absent in chlorophyll C. That is the thing which is seen in case of the particular this different type of chlorophylls. Okay, so in this chlorophyll A is important that we have discussed. Chlorophyll B, C have some important points that also we have discussed. That is all about the what chlorophyll. Now after this second group of pigment we are going to discuss, that is carotenoids. And if we talk about carotenoids, they are yellow to yellow orange color pigment. They are yellow to yellow orange color pigments, and that occur inside chromoplast, that occur inside chromoplast, and also occur along with chlorophyll, occur along with chlorophyll inside chloroplast. So they are also found in chloroplast as well as chromoplast. Okay, chromoplast are the colored plastid and chloroplast are the greenish plastid. This pigment carotenoids are considered as universal in occurrence and insoluble in water. They are insoluble in water, obviously. Okay, and they are most stable pigment, considered as a most stable pigment, and light is not necessary for their synthesis. In chlorophyll light is necessary, but here light is not necessary for their synthesis. And it is considered they are a hydrocarbon with the conjugated double bond, and it is considered that these carotenoids are of two type. Carotenoids are of two type. Carotenoids are of two type, and in this carotenes are there and the xanthophyll are there. Now carotenes are a yellow orange colored pigment. They are yellow orange colored pigments, purely hydrocarbon, and the empirical formula is C40H56. Okay, they are of three types. They can be alpha, beta, gamma carotene, and beta carotene are common in plants. Out of this, beta carotene are common in plants and also responsible for and also responsible for color of carrot. Now apart from this, the next carotenoids are the xanthophyll, and these are considered as yellow colored oxygen containing oxygen containing derivatives of carotenes. And their formula can be C40H56O, C40H56O2, etc. And in that, it is known that they are of different types. And if we talk about the types of xanthophyll, they are a viola, xanthine, They are zeaxanthin, fucoxanthin, diatoxanthin, dinoxanthin. These all are particularly different types of xanthophylls. In that, we remember that lutein is a type of xanthophyll. Lutein is a xanthophyll which is common in plants, and responsible for autumn color of leaf. Autumn color of leaf. Now, if we talk about the functions of carotenoids, then it is known that if we talk about the functions of carotenoids, then they are accessory pigment, and if they are accessory pigment, they make photosynthesis more efficient by absorbing different wavelength of light, and they provide ultimate energy to the reaction center. Apart from this, these carotenoids protect chlorophyll A from photooxidation. From photooxidation. So they protect the photosynthetic machinery, means chlorophyll A, by converting lethal method oxygen into the unharmful. So photooxygen from photooxygen by converting lethal nascent oxygen, lethal nascent oxygen into unharmful molecular oxygen, thus called shield pigments. Okay. Apart from this, it is also considered as beta carotene is precursor of vitamin A. Now the question is how it is getting synthesized. So if we write a reaction, dear student, beta carotene is C40H56. If we react it with the two water molecule, it is going to form two molecule of C19H27 CH2OH, that is vitamin A. This way the vitamin A is synthesized. And apart from this, carotenoids help in entomophily and zoochory. Now, entomophily is the pollination by insects, and zoochory is the seed dispersal by animals. So, in this way, to the flower, okay, they give a particular color that is due to the carotenoids. So, this way, carotenoids has been these two mechanism. Okay, so this is all about the carotenoids. Apart from this, the third group which we have, that is phycobilins. Now those phycobilins are hot water soluble pigment. they are hot water soluble pigments. Okay, and they are found to be associated with protein. They are not found singly, they are found to be associated with the protein. Apart from this, they lack magnesium and phytol tail compared to chlorophyll A. So they are open tetrapyrrole pigments, means they have pyrrole ring, four pyrrole ring, but this open four pyrrole ring are ultimately forming a phycobilins. And if we talk about the types of phycobilins, they are phycocyanin, which is blue in color. Second, phycoerythrin, that is red in color. And third, yellow phycocyanin, that is light blue in color. So these all are the phycobilins. So this phycobilins, if we talk about location, these phycobilins occur exclusively in blue-green algae. They are found or occur exclusively, means only in blue-green algae and red algae. So they are exclusively found occur in blue-green algae and red algae. Okay, as an accessory pigment. Okay, so that is the thing with respect to the phycobilins. Okay, so that is what about the different pigments. Okay, now after discussing pigment, if we have discussed the pigment, we have to discuss two more things which also help the TW Engelmann to carry out their experiment, and that is the absorption and action spectrum that we have to understand here. So now we are going to discuss absorption spectrum and action spectrum. Absorption spectrum and action spectrum. These two things we are going to discuss here. So first, if we talk, this is the absorption spectrum. Now, what is this absorption spectrum? So it is nothing but it is a graphic representation. It is graphic representation. There is a graphic representation of absorption of different wavelength, of absorption of different wavelength, of different wavelength of light by various pigment molecules. By various pigment molecule, that is chlorophyll A, chlorophyll B, and chlorophyll B. So if we draw a curve, that is absorption spectrum, here we are taking a on x-axis, that is a wavelength of light in nanometers. That is, this is the 400 wavelength, this is 500, this is 600, this is 700 nanometer. This white line showing the nanometer wavelength. And on the y-axis, we are taking an absorption of light by a chloroplast pigment. So when we see the absorption, This dark color shows the chlorophyll A. Sorry, this chlorophyll A is this one. This one is showing the chlorophyll A. So chlorophyll A peak you will see near about 430, 440, and here also you will see the chlorophyll A peak near it is about near 660. Same way chlorophyll B peak also are observed around 665 or 670, and chlorophyll B peak are there near about 650. When we talk about the carotenoids peak, carotenoid maximum absorption peak means maximum absorption of that particular wavelength occurring. So one, two peak are observed at 450 to 500. So that's the maximum absorption. Then the chlorophyll, then carotenoid cannot show absorption beyond 520 wavelength. Okay. So this is the how the peak are shown for this three pigment. That is peaks are showing the particular wavelength, how much that particular wavelength is getting absorbed. So this is the absorption spectrum when we draw a curve of this three pigment with the absorption with respect to the wavelength. Now, this is the action spectrum. This is the action spectrum. So, after absorption, what is the action? The action is photosynthesis, and photosynthesis is, means the what? A food synthesis, and in food synthesis, along with the food synthesis, what will happen? The oxygen release rate will come. So, in action spectrum, we take a wavelength on the particularly the x-axis, that is 400, 500, 600, and 700. So in action spectrum wavelength, that is 400 to 700 is taken. Now, after absorbing light, what is the action? Action is the food synthesis, and food synthesis, what happens? Oxygen evolution occur by the CO2 friction. So rate of photosynthesis is measured by the oxygen release. So the maximum oxygen release was observed at this peak, and this peak means at this wavelength, that is about 430 near about, and at the near about 700. So this is the action spectrum, that this much oxygen evolution observed along with this wavelength. Now, when they particularly shown a resemblance or compared this both the curves of absorption spectrum and action spectrum, then this was observed. So it is a comparison of absorption and action spectrum. Now in this comparison, you can see this dark black color shows the rate of photosynthesis, that is action spectrum. And when we see the matching of a comparison of this action spectrum with the absorption spectrum, this curve of absorption spectrum matches mainly with this, the rate of photosynthesis. And this curve is of a chlorophyll A. So from this, it is understood that the chlorophyll A resemblance with the absorption spectrum. Actually, chlorophyll A and chlorophyll B both resemble, but the chlorophyll A is showing a maximum resemblance. So the chlorophyll A is the main pigment that can be understood from here. So chlorophyll A is the main pigment. So absorption spectrum we have discussed, action spectrum we have discussed, and ultimately we have shown that here chlorophyll A and B showing resemblance with action spectrum, but in this the chlorophyll A is the main photosynthetic pigment. A is main photosynthetic pigment because maximum resemblance with the action spectrum is of chlorophyll A action absorption spectrum. So chlorophyll A is considered as main photosynthetic pigment, and that is absorption action spectrum, and that is all about the pigment. So the main photosynthetic pigment in plant photosynthesis is chlorophyll A, just we have discussed. Photosynthetic active radiation PAR represents the following range of wavelength. It is showing a 400 to 700 nanometer is considered as a photosynthetically active radiation which is used in absorption and action spectrum. Accessory pigments include chlorophyll B, xanthophyll, carotenoid, all are considered as accessory pigments. So all will be the correct answer. Okay, so that is all about the different pigments, the photosystem, location, how these all are located, that all thing we have discuss. Okay, so I hope you understood well. Thank you. From next class, we will be discussing light reaction and dark reaction in detail.